Welcome back to our channel. Today we are going to study about biological classification. Topics that we are going to cover today are classification, two-kingdom system, modern classification, salient features, five-kingdom classification, three-domain system and Kingdom Monera. This video is the first part of biological classification. Stay tuned for the upcoming second part, which will be released shortly. Kindly refer this page for the syllabus and previous year questions topics. Introduction to classification. Humans have classified organisms since ancient times. Early classification was instinctive, based on utility, food, shelter, clothing. No scientific criteria were used initially. Aristotle's contribution. First scientific approach to classification. Classified plants into trees, shrubs, and herbs. Divided animals into 1. With red blood, vertebrates, 2. Without red blood, invertebrates. Linnaeus and the two-kingdom system. Introduced the two-kingdom classification. 1. Plantae, 2. Animalia. Included all plants and animals respectively. So, these are the limitations of the two-kingdom system. No distinction between prokaryotes and eukaryotes, unicellular and multicellular, photosynthetic and non-photosynthetic organisms. Example, green algae and fungi were misclassified. Why change was needed? Many organisms didn't fit into the plant or animal kingdom. So, two-kingdom system became inadequate. Basis for modern classification. Needed more characters beyond morphology. 1. Cell structure, 2. Nature of the cell wall, 3. Mode of nutrition, 4. Habitat. 5. Reproduction, 6. Evolutionary relationships. Evolution of classification systems. Classification has evolved with scientific discoveries. Constant kingdoms are Plantae and Animalia. But the number and definition of kingdoms have varied with time. So, with this we come to a conclusion that 1. Classification started for utility, evolved with science. 2. From two kingdoms it is now five kingdoms and beyond, Whitaker, Woes. 3. Scientific classification now uses cell structure, genetics, evolution, nutrition and reproduction patterns. I am going to explain this table in detail. Characteristics of the five kingdoms. 1. Cell type, Monera is prokaryotic, no nucleus or membrane-bound organelles. Protista, fungi, Plantae, Animalia are eukaryotic, have a nucleus and membrane-bound organelles. 2. Cell wall, in Monera, cell wall is present but made of non-cellulosic material, polysaccharides and amino acids. In Protista, cell wall is present in some, not all, varied composition. In fungi, cell wall is present, made of chitin. In Plantae, cell wall is present, made of cellulose. But in Animalia, it is absent, animals do not have a cell wall. 3. Nuclear membrane in Monera, nuclear membrane is absent, hence classified as prokaryotes. But in Protista, fungi, Plantae, Animalia, nuclear membrane is present, confirming they are eukaryotes. 4. Body organization, Monera and Protista, has a cellular level of body organization, mostly unicellular. Fungi, has a multicellular or loose tissue structure. Plantae, have tissue and organ-level organization. Animalia, has the most complex with tissue, organ, and organ system-level organization. 5. Mode of nutrition, Monera, can be autotrophic, can be chemosynthetic, use chemicals, or photosynthetic. Heterotrophic, saprophytic, feeds on dead matter, or parasitic. Protista, can be autotrophic, photosynthetic, like algae. Heterotrophic, example, protozoa. Fungi is heterotrophic, mainly saprophytic or parasitic. Plantae is autotrophic, perform photosynthesis. Animalia is heterotrophic, can be holozoic, ingest solid food, saprophytic, etc. Introduction to Five Kingdom Classification. R. H. Whitaker, 1969, proposed the Five Kingdom Classification. The Five Kingdoms are 1. Monera. 2. Protista. 3. Fungi. 4. Plantae. 5. Animalia. Based on criteria like 1. Cell structure. 2. Body organization. 3. Mode of nutrition. 4. Reproduction. 5. Phylogenetic, evolutionary, relationships. Evolution of classification. Early classification grouped all organisms with a cell wall under plants. Included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and angiosperms. This system didn't consider prokaryotic and eukaryotic cells, unicellular and multicellular forms, autotrophic and heterotrophic nutrition. Problems in old systems. Chlamydomonas is unicellular, and Spirogyra is multicellular but were placed in the algae. Fungi and green plants were grouped despite. Fungi are heterotrophic and has chitin wall, and plants are autotrophic and have cellulose wall. Hence, there was a need for a better classification system. Whitaker's contribution. Whitaker separated organisms based on true characteristics. Placed all prokaryotes into Kingdom Monera. And all unicellular eukaryotes into Kingdom Protista. Fungi has its own kingdom due to heterotrophy and chitin walls. Kingdom Protista. Why is it needed? Grouped organisms like Chlamydomonas, chlorella had a cell wall and are autotrophic. Amoeba, Paramecium has no cell wall and are heterotrophic. These were previously split into the plant and animal kingdoms. Protista unified them under unicellular eukaryotes. Importance of modern classification. Classification criteria evolved with better understanding. Modern systems try to reflect not just morphology or reproduction, but also evolutionary, phylogenetic relationships. Three-domain system. A newer system, three-domain, divides Monera into 1. Archaea. Ancient, primitive microorganisms. Live in extreme environments like hot springs, salt lakes. Cell walls without peptidoglycan. More closely related to eukaryotes than to bacteria. 2. Bacteria. True bacteria, also called Eubacteria. Found everywhere from soil, water, air, and in organisms. Cell wall made of peptidoglycan. Includes autotrophs, make their food, and heterotrophs. 3. Eukarya, includes Protista, fungi, Plantae, Animalia. We will study more in higher classes. Organisms with a true nucleus and membrane-bound organelles. Includes 4 kingdoms. Protista that are mostly unicellular eukaryotes. Fungi are heterotrophs with chitin cell walls. Plantae are autotrophs with cellulose cell walls. Animalia are heterotrophs with no cell walls. In this chapter we will deal with the characteristics of 1. Kingdom Monera. 2. Kingdom Protista and 3. Kingdom Fungi. Kingdom Plantae and Animalia will be covered in later chapters. Introduction to Monera. Includes only bacteria, the most abundant and ancient microorganisms on Earth. It is found everywhere: soil, water, extreme environments, hot springs, deserts, snow, deep oceans. Can be free-living, symbiotic, or parasitic, living in/on other organisms. Bacterial shapes. Bacteria are classified based on shape into 4 types. 1. Coccus, cocci, round-shaped, bacilli, cylindrical or rod-shaped, have a spore to survive in extreme environments. Spirillum, spiral-shaped, vibrio, comma-shaped. Cell structure. Prokaryotic, no nucleus or membrane-bound organelles. Surrounded by a rigid cell wall. DNA is naked and lies in the nucleoid region. Some have flagella for movement and pili for attachment. Metabolic diversity. Bacteria show extraordinary metabolic adaptability. 1. Autotrophic bacteria. First, photosynthetic autotrophs, use sunlight to make food, example, cyanobacteria. Second, chemosynthetic autotrophs, derive energy by oxidizing inorganic substances, example, nitrifying bacteria. 2. Heterotrophic bacteria, depend on organic substances from other organisms. Can be saprophytic, feed on dead matter, or parasitic, feed on host. Key features. Features and their description. 1. Cell type of Monera, it is prokaryotic. 2. Cell wall is present in Monera, it contains peptidoglycan. 3. Nutrition autotrophic/heterotrophic. 4. Reproduction in Monera is mostly asexual by binary fission. 5. Habitat of Monera is ubiquitous that it is present everywhere. Why is Monera important? Ecological role, they are decomposers, nitrogen fixers. Economic value, they are used in fermentation, antibiotics, and biotech. Pathogens, some cause diseases in humans, animals, and plants. Introduction to Archaebacteria. What are Archaebacteria? Archaebacteria are a unique group of primitive prokaryotic organisms. It can survive in extreme environmental conditions where most life forms cannot. Habitat, extreme environments. They are called extremophiles because they thrive in harsh conditions such as in hot springs, they are Thermoacidophiles. In salty areas, they are Halophiles. In marshy environments/guts of ruminants, they are Methanogens. Special features are: Archaebacteria lack peptidoglycan in their cell wall unlike other bacteria. Instead, they have unique cell wall lipids that provide resistance to high temperature, acidity, and salinity. This distinct cell wall composition helps them survive extreme conditions. Examples and importance of Methanogens. Found in the gut of ruminants. Example: cows, buffalo. Helps in digestion and releases methane gas. Play a key role in biogas production from dung. Key features of Archaebacteria. Key features and description of Archaebacteria. Habitat of Archaebacteria is they survive on extreme, hot springs, salty areas, marsh. Examples are Halophiles, Thermoacidophiles, Methanogens. They have a unique cell wall which contains no peptidoglycan, it is resistant to lipids. Their role in biogas is Methanogens produce methane in ruminants. Why is Archaebacteria important? Survive extreme conditions, heat, salt, no oxygen. Produce biogas, methane, in ruminant animals' guts, example, cows, buffaloes. Used in biotechnology, enzymes stable at high temperatures, example PCR, polymerase chain reaction. Help understand evolution, among the earliest life forms. Play ecological roles in the carbon and nitrogen cycles. Mnemonics down there, you can refer it for Kingdom Monera. If it is helpful, you can comment the mnemonic so that we get to know it was helpful for you. Or if you found it difficult, you can use simplest one from somewhere if you want. Introduction to Eubacteria, true bacteria. General features of Eubacteria. Known as true bacteria. Possess a rigid cell wall. Modal types have flagella. Diverse in structure and function. 1. Cyanobacteria, blue-green algae. Have chlorophyll like green plants, they are photosynthetic autotrophs. Forms, unicellular, colonial, or filamentous. Habitats are freshwater, marine, terrestrial. Colonies are enclosed in a gelatinous sheath. Cause blooms in polluted water bodies. Some, example, nostoc, anabina, fix nitrogen in heterocysts. 2. Chemosynthetic autotrophic bacteria. Obtain energy by oxidizing inorganic substances such as nitrates, nitrites, ammonia. Use this energy to make ADP. They play roles in recycle nitrogen, phosphorus, iron, and sulfur. 3. Heterotrophic bacteria. Most abundant in nature. Functions are: decomposition means break down organic matter. Useful for humans such as make curd from milk. Produce antibiotics. Nitrogen fixation in legume roots, rhizobium. Some are pathogens, cause diseases like cholera, typhoid, tetanus, citrus canker. Reproduction in bacteria. Mainly by binary fission, asexual. Under harsh conditions, they form spores. Can exchange DNA via primitive sexual reproduction, example conjugation. Mycoplasma. Do not have cell wall so they are unique. Smallest living cells. Survive without oxygen, anaerobic. Many are pathogenic to plants and animals. Why is Eubacteria important? 1. Nutrient recycling, decompose organic matter and recycle nutrients like nitrogen and carbon in ecosystems. 2. Nitrogen fixation, convert atmospheric nitrogen into usable forms for plants, example rhizobium, anabina. 3. Photosynthesis, cyanobacteria produce oxygen and support aquatic food chains. 4. Industrial uses, used in making curd, cheese, alcohol, vinegar, and antibiotics. 5. Medical importance, source of medicines, antibiotics, but also causes diseases like cholera, typhoid, and tuberculosis. 6. Scientific research, E. coli and others are widely used in biotechnology and genetic engineering. Bonus tip is down there. Comment bonus tip if you like it to get more like this. Key features of Eubacteria. Key features and their description of Eubacteria. 1. Cell wall is made of peptidoglycan, muriin, gives shape and protection. 2. Flagella is present in motile bacteria, helps in movement. 3. Cyanobacteria, blue-green algae, contain chlorophyll A, perform photosynthesis, may fix nitrogen in heterocysts, found in fresh/marine water. 4. Unicellular or colonial may exist as single cells, colonies, or filamentous forms. 5. Autotrophs are either photosynthetic, like plants, or chemosynthetic, oxidize inorganic substances for energy. 6. Heterotrophs mean they are dependent on other organisms for food, include decomposers and pathogens. 7. Reproduction is mostly by binary fission, spore formation under unfavorable conditions, may show primitive DNA transfer. 8. Mycoplasma has no cell wall, smallest living cells, survive without oxygen, anaerobic, often pathogenic. Thanks for watching. If you found it helpful, like, share and subscribe. Follow us on our socials so that you get every update. Do not miss anything. You will get the notes from the Linktree. We will see you in next video. Stay tuned for the upcoming second part, which will be released shortly.